says that just as a construction crew uses blueprints to build a house, a cell uses DNA as plans for building proteins. In addition to DNA, another nucleic acid called RNA is involved in making proteins. In this gizmo, you will use both DNA and RNA to construct a protein out of amino acids. DNA is composed of the four bases, A, C, G, and T. RNA has A, C, and G, but then the U gets replaced with T, which we just reviewed. So when I open the simulation, you're going to look at the simulation pane right here. And is the displayed segment part of a DNA or RNA molecule? So if you look right here, is this DNA or is that RNA? Just based on its letters. If we have A, T, C, and G, because there are T bases, not U bases. All right, number 2. RNA polymerase is a type of enzyme. Remember, anything ending with ASE is an enzyme. Enzymes help chemical reactions occur quicker. So we are going to release the enzyme, and you're just going to describe what happens. So here I am in Gizmo. I already went to ClassLink and opened it up. Full screen, here is my RNA polymerase. I'm going to click release. And what happens? It splits, or it gets separated. So for number 2, you can just say it splits apart, it gets separated. Okay, so we already clicked release enzyme. All right, so the first stage of building a protein involves a process known as transcription. In transcription, segment of DNA serves as a template, or the original copy, to produce a complementary strand of messenger RNA, which just is also called mRNA. So we're basically going to be building it. Like DNA, RNA follows the base pairing rules. So A, apples and trees, cars in the garage, except remember that instead of a T, there's a what? Because this is question 1. Oh, sorry. Which RNA base bonds with T? So what is in a tree? So you can write A, or if you remember the whole word, adenine. I don't care about spelling. All right, so here is where I want to know what replaces T, because remember apples and trees, but now we're talking about RNA. So what base is going to pair with A now? What letter? You can just tell me the letter. Okay, it would be thymine if we're talking about DNA. So then, perfect. The T gets switched to a U, which is uracil. Which that is the question for number 3. In a molecule of RNA, uracil takes the place of the DNA base thymine. The rest are the same. And then we're going to go over how to build it. So back to our gizmo, we have to build our messenger RNA. So you guys told me that A pairs with T, so I'm going to put the A next to the T. You told me that uracil U gets switched with a T, so that's going to go there. Cars in the garage. Cars in the garage. It would be apples in the tree, but I get a U instead of a T. Oops. So once you build your sequence, oops, that goes down here. Can I do that? Yes, I can. Once you have your sequence, the next question is just to write this down. So on number 4, it says, what is the nucleotide sequence of your messenger RNA? It says this sequence right here, AUG, CUG, ACC, UAG. Can I remember that? ACC, UAG. All right, so once you click continue, when you're on activity B, so after a strand of mRNA has been built, which you did, the strand exits the cell's nucleus and it goes out into the cytoplasm where it finds a ribosome to build the chain of amino acids to make a protein. All right, so we're going to look at the strand of mRNA, and every group of 3 is called a codon. So we're going to just write down the 3 bases that make up our codon first. So the first 3 letters that make up a codon is AUG. So that's our first one. So on our Gizmo, you just write AUG. Our second pair of 3 letters is CUG. Oh, sorry. There you go. Then we'll read the next 3 pairs is ACC. And our last 3 down here at the bottom is UAG. Okay, so then translation starts when the ribosome, or the purple structure in your Gizmo, binds or connects to the strand of mRNA. Remember, tRNA, or the truck, has the amino acid carrying it around. So if you think of like a dump truck, it's carrying around the amino acid in the back. And each tRNA molecule carries only one kind of amino acid. So the amino acid is determined by the anticodon, or reading these bases. So our first question is, which anticodon, or what bases, are going to attach to this first one, AUG? So you would just read it again backwards. So what pairs with A would be a U, what pairs with U is an A, and what pairs with G is cars in the garage. That one stays the same. And so that's what we're going to try and find on our Gizmo, UAC. So I found UAC on my truck, or my transfer RNA, this little green guy over here. I connect it to this one because that's the anticodon, or the opposite, and it matches. So then it says continue to match the truck RNA anticodons to create a protein. So what connects with a C is a G. This one starts with a G, so I know that that one's going to be next. Grab that, put that there, and look, it moves along. So then the ribosome keeps reading. What matches with an A? Usually it would be a T, which means it switches to a U. Put that one on there, moves along. And then there's no other tRNAs, so we just know that the release factor comes next. Just plop that on the back end, and we've created a small protein.